On this slide, we're looking at a diagram of the process of facilitated diffusion. So we've got a representation of a membrane in the middle of the slide. You can see heads and tails and heads, the membrane bilayer. You can also see these structures drawn in. Two transmembrane proteins. Remember, these are integral membrane proteins, and because they cross both monolayers in the membrane, we can call them transmembrane proteins as well. This one represents a channel protein, and this one represents a carrier protein. Those are the two types of proteins that perform facilitated diffusion. Now, we've got two different solute molecules drawn in this diagram. We've got this yellow circle and we've got this pink triangle. So these are whatever you might imagine they are. They're molecules that cannot cross a membrane by simple diffusion. So they need some help, they need some assistance from proteins. That's what facilitated diffusion is all about. The yellow molecule has a concentration gradient across this membrane. There's lots more yellow out here than there is in here. Let's imagine that this is the outside of the cell and this is the inside of the cell. The pink molecule also has a concentration gradient across this membrane. There's more pink triangles outside the cell than there is inside the cell. Let's start with the channel protein first. There's a concentration gradient in place. That's all we need in addition to the protein for facilitated diffusion to occur. We just need that gradient in place. We don't need to put any energy into facilitated diffusion. In other words, the cell doesn't have to spend any energy to make this happen. It's the fact that there's a concentration gradient. That is what's going to make it happen. And it's going to happen with the help of this channel protein. The yellow circles are going to move from the side with the high concentration through the protein to the side with the low concentration. The protein is shaped in a way that gives it a literal tunnel, a channel through the middle. Now they've cut it in half for us here, but you have to imagine it in 3 dimensions. It's a tube, it's a pipeline, or a tunnel, whichever analogy you like. When you cut it in half, you can see that space in the center. That's where the yellow molecule is traveling. This yellow molecule, it has something about it that prevents it from simply crossing the phospholipids. Maybe it has a charge on it. Maybe it has a molecular weight that's higher than 100. Or maybe it's relatively polar. Whatever the case may be, it can't use simple diffusion to get across a membrane, so it's going to use facilitated diffusion instead. And it's going to travel from the high concentration side through the protein towards the low concentration side. And it will keep flowing through that channel protein until the gradient is gone, until there are equal amounts of yellow circle on both sides of the membrane. Now take a look at the carrier protein, because this one functions quite differently. It turns out that channels and carriers are specific. In other words, this channel is only going to work for the yellow circle. If the pink triangle, for example, tried to come through this channel, it wouldn't work. It wouldn't fit through that channel. A similar situation exists with carriers. They are specific. So if the yellow circle tried to pass using this carrier, it just wouldn't work. This carrier only functions for the pink triangle. So we have a high concentration of pink triangles on the outside and a low concentration on the inside. That means facilitated diffusion is going to move this way across the membrane. It's going to these pink triangles are going to travel using the carrier protein from the outside of the cell to the inside of the cell. And that will keep happening until the concentration of pink triangle is the same on both sides. Now, how does this protein function? Well, this carrier would be open to pink triangles entering right here. You have to use your imagination a little bit here, but carrier proteins are open on one side and closed on the other side in their resting state. The pink triangle, or whatever molecule this carrier is specific for, is going to not only be able to come in, but there's going to be a place inside the protein that fits this molecule perfectly. So only the pink triangle will be able to come in through the open side and fit in this protein the way a key fits in a lock. When the pink triangle comes in and binds to that place, and again, think of it like a key going into a lock, that's going to cause this protein to change shape. Remember, proteins are folded up in one particular way so that they can form one particular function. If you add another molecule to a protein, you're going to cause that protein to refold slightly, to change its shape. So when the pink triangle comes into this opening and sits right here at this particular site in the protein, it will interact with the protein and cause it to change shape. That shape change will close the opening over here and open the protein over here. That will allow the pink triangle to now pass out on this side. When the pink triangle leaves the protein, in other words, when it stops interacting with it, the protein is going to go back to its original shape. Open on this end, closed on this end. Another pink triangle can come in through the opening and bind right here. The binding action will cause the protein to change shape. It will close up over here and open up over here. The pink triangle can then come out and the protein will go back to its original shape. So carrier proteins can function again and again and again. They can carry pink triangles or whatever molecule they're carrying across the membrane until the concentration gradient is gone, and then facilitated diffusion will stop. Remember, facilitated diffusion and simple diffusion are spontaneous. They don't require any energy to make them happen. All they require is a concentration gradient. The last transport mechanism is different. The last transport mechanism, active transport, does require energy. And the reason it requires energy is because it's going to move a solute, a dissolved molecule, against its concentration gradient. It's going to move it in a direction that keeps the gradient in place. It doesn't help the gradient go away. It keeps the gradient in place. Nature doesn't want that gradient to stay in place. Nature wants it to resolve. So in order to keep it in place, the cell is going to have to invest some energy to make that happen. And that's what active transport is all about. Now, active transport does share one thing in common with facilitated diffusion, and that's that it requires a protein. We call these active transport proteins pump proteins. Now, if you look at the diagram on the slide, if you start up at the top, what they're showing us in this picture is the direction of active transport. There's a concentration gradient drawn in this picture. There's more red circles on this side, there's fewer red circles on this side, and there's a black membrane in between. So if we expected simple diffusion or facilitated diffusion to occur, we would predict that the red circles would travel this way. They would go from the side where the high concentration is, across the membrane to the side where the low concentration is. But this is active transport, and active transport goes in exactly the opposite direction. Active transport is going to transport red circles from the side where there's a low concentration toward the side where there's a high concentration. In other words, it's going to keep this gradient in place. Now you might ask yourself, what happens when there are no more red circles over here? Well, what happens in a real cell with a real membrane is that some of these red circles are going to get across this membrane in this direction. That's a phenomenon that we call leakage. And we're going to talk more about leakage when we discuss muscle physiology and nerve physiology. But for now, just understand that the natural drive for a concentration gradient to go away is so strong that some molecules are going to leak this way across the membrane. So active transport keeps going all the time, 24 hours a day, 7 days a week, as long as the cell has enough energy to keep it going. There's never a point where the pump protein can stop working, in other words, because of this phenomenon of leakage. Now, down at the bottom, this is simply another artist's depiction of active transport, and you can see in this drawing, it's going in the other direction. The idea is the same, though. There's a concentration gradient in this drawing. There's a high concentration on this side. This represents the inside of a cell. And there's a low concentration on this side. This represents the outside of a cell. And there's a membrane in the middle. And this purple membrane protein, which happens to be a pump protein. This pump is going to carry the green squares from the side where there's a low concentration towards the side where there's a high concentration. It's going to keep this gradient in place. And the only way it can manage that is by spending energy. And the energy molecule, the currency in the cell, is ATP. So it will break apart this ATP, it will break a covalent bond, and get a little burst of energy that will fuel this pump. So as you're studying these three transport processes, simple diffusion, facilitated diffusion, and active transport, it's a good exercise to ask yourself what things they have in common and what things they don't. When we think about simple diffusion and facilitated diffusion, the things they have in common are the fact that they are spontaneous, the fact that no energy needs to be invested to make them occur. As long as there's a concentration gradient across the membrane, these two processes will occur. Now, the difference between them is that simple diffusion happens between the phospholipids, and facilitated diffusion requires a protein, either a channel or a carrier. Active transport is very different. Active transport requires energy. It is a non-spontaneous process. It's going to move molecules up the concentration gradient or against the concentration gradient. In other words, it's going to maintain the gradient. Now, it does have one thing in common with facilitated diffusion in that it requires a membrane protein, and it's a particular protein called a protein pump. Now, the last thing you'll notice in the drawing is that because we've called this active transport, and again, this is the mechanism that requires energy, the artist has noted that these two are passive transport. All that terminology means is that they don't require energy. Simple diffusion, facilitated diffusion, they are passive. They don't require a source of energy to make them happen. They simply require a concentration gradient. They will keep happening as long as the concentration gradient is in place. Once equilibrium is achieved, they will stop. Active transport is different. Active transport maintains a concentration gradient. In this drawing, you can see there are relatively few yellow diamonds on this side and more yellow diamonds on this side. So there's a concentration gradient. And this pump is moving yellow diamonds from the side with the low concentration towards the side with the high concentration. It's keeping the gradient in place. In order to do that, it needs a source of energy. And that source of energy is ATP.